conversion of pyruvate to acetyl-CoA, and then through TCA cycle. So let's go ahead to our presentation again. I will share my screen. Can you see the screen?
Yes, sir.
Good. Yesterday we had a discussion on this issue of citric acid cycle, and that is the last component where the acetyl fragment, that is a 2-carbon fragment, is completely oxidized at 2 locations where the carbon is given out in the form of CO2, and at 3 locations there is an oxidation of these organic acids. And when these organic acids are undergoing oxidation, obviously there is a formation of NADH2, a reducing compound. At one stage there is a synthesis of FADH2, flavin adenine dinucleotide, and at one location from succinyl-CoA to succinate, there is a synthesis of GTP from GDP, and ultimately GDP is giving out the phosphate group to ATP, and as a result we are getting the ATP at the end. So to begin with, what is the net gain of TCA cycle? The 2 molecules of carbon dioxide, that is CO2, that is decarbonization process. The 3 molecules of NADH2, as I told you now, we have discussed. One is from isocitrate to alpha ketoglutarate, from alpha ketoglutarate to succinyl-CoA, and lastly from malate to oxaloacetate. And there is one component from succinate to fumarate, we are going to get the FADH2. So obviously we are going to give the 3 hydrogen ions which are being undertaken, which are taken out. Each glucose molecules, obviously when it produces the 2 pyruvate molecules, which in turn they are producing the 2 molecules of acetyl-CoA, and then acetyl-CoA is undergoing this whole TCA cycle or citric acid cycle. It means that for every glucose molecule, this cycle takes place 2 turns. So obviously we have to double these oxidation steps. So here, what is the importance of respiration? The importance of respiration is that here the sugar and the organic acids, they are oxidized. During their oxidation, the energy is given out, and this energy is trapped by NAD molecule, and the NAD is converted into NADH2. So NAD is reduced to NADH2, and so there are, you can see, almost there are 7 steps in the total complete oxidation of glucose into its CO2 and water. Obviously the first step is 3-PGAL to 1,3-diphosphoglycerate, aldehyde to glycerate. So this step is the first step which is taking place in the glycolysis. Pyruvate to acetyl-CoA, this step is taking place in the stroma of the mitochondrion, where the acetyl-CoA is synthesized, and then the acetyl-CoA enters into the TCA cycle or citric acid cycle, and these are the 5 steps, of which the 4 steps are providing, the 3 steps are providing the NADH, 1 step is providing the FADH2, and 1 step is providing the ATP molecule. So isocitrate to alpha ketoglutarate to succinyl-CoA, succinyl-CoA to succinate to fumarate, and ultimately malate to oxaloacetate. So these are correspondingly providing us the NADH2, FADH2 and GTP molecule or ATP molecule. Now when this NADH2 is synthesized, ultimately the energy which is present in binding these hydrogen protons, that is utilized for the synthesis of ATP molecule. And that's why here another oxidation reduction mechanism is present, which is involving the iron and which is involving the cytochromes, a specific form of proteins and nitrogen compounds, and these are undergoing the oxidation and reduction by handling over the electrons which are released during this process of oxidation. And during this process, when the electrons are passing out, they are releasing the energy, and this energy is trapped along with the hydrogen protons which are released now for the synthesis of ATP molecules. And that's why this another component or the next component is known as an electron transport system, or it is also called as a terminal phosphorylation, or it is also called the oxidative phosphorylation. It is called the oxidative phosphorylation because here it is taking place in the mitochondrion, and at the end the oxygen is involved, which is responsible for generation of high energy phosphate bond. When we talk about this chain, this is also called as a cytochrome chain, or it is also called as the flow is called as the electron transport system. Now there are four distinct sites which are involved. The first site, as you can see in the image, it contains a fragment of polyphenolic compound FMN, that is flavine mononucleotide, and a cytochrome Q. They are forming one separate site. The next site involves a chain of cytochromes where the cytochrome B, cytochrome C1 and cytochrome C are involved. They are passing the electrons one by one. And last is called as the terminal oxidase enzyme, or it is involving the cytochrome A and cytochrome A3. Remember, cytochrome A and cytochrome A3, along with the iron, they are also containing the copper, and that's why these are specific terminal oxidase enzyme system which are responsible for joining this electron with the hydrogen proton to form a hydrogen ion, and this hydrogen ion ultimately links with the atmospheric oxygen so as to form the water molecule. Now, as I told you, this image shows the site 1, site 2 and site 3, but this is for the sake of convenience. Actually, the site 1 is shown here. The site 2 is the below site, that is FAD. The site 2 is actually the site 3, and site 3 is actually the site 4, that is a terminal oxidase enzyme. Now, what is happening here? The NADH2 which is synthesized, this NADH2 ultimately gives out the hydrogen proton, and it is again oxidized to NAD. When it undergoes oxidation, it gives out the electron to FMN, and this FMN accepts the electron and the hydrogen proton, and it gets reduced. And in this way, the oxidation reduction takes place in the FMN, cytochrome Q, cytochrome B, cytochrome C1, cytochrome C, and ultimately the last terminal oxidase, that is cytochrome A and cytochrome A3. And in this way, in a chronological fashion, the electron passes through these cytochromes, and this energy lost electron is ultimately again accepted by the hydrogen protons, which are released at the cytochrome Q level, and this hydrogen proton accepts the electron to form the hydrogen ion. The hydrogen ion ultimately joins with the oxygen to form the H2O molecule. Now you can see the beauty is that in between Fmn and Q, cytochrome Q, this energy drop is much more higher, and that's why the cytochrome, the site 1 is responsible for giving out the sufficient energy to generate one ATP molecule. Similarly, site 2, that is cytochrome B3 to cytochrome C1, this site is also having a sufficient redox potential drop so as to again synthesize one molecule of ATP. And lastly, the site 3, which is the terminal oxidase, it is also having a sufficient redox potential drop of the electron energy so that it also generates one more ATP molecule. So in this way, at three locations, there is a sufficient energy drop of the electrons, and because of that, this energy and the hydrogen proton influx, that is movement of the hydrogen protons across the membrane, is responsible for the synthesis of ATP molecules. How these ATP molecules are being synthesized, we'll be studying it again. But remember that there is only one enzyme reaction, that is succinate to fumarate, where FAD is involved, and this FAD, when it gets reduced to FADH2, this FADH2 again gets oxidized, releasing this hydrogen proton, and this hydrogen proton is accepted by the cytochrome Q. But remember, during this whole process, this particular oxidation reduction reaction is having much more less energy, and because of that, when FADH2 gets oxidized, only 2 ATP molecules are synthesized. So what is the beauty of this reaction sequence or the electron transport system? That when NADH2, the reduced compound, when it gets oxidized, the 3 ATP molecules are synthesized because of the 3 locations where the energy transfer is taking place. Whereas when the succinate is undergoing oxidation to fumarate, and when the FAD is getting reduced to FADH2, then the FADH2, when it is entering into this electron transport system, it is releasing less amount of energy, and because of that, there is a synthesis of only 2 molecules of ATP. And that's why this is a diagrammatic presentation of cytochrome chains, which is also known as an electron transport system. Dear friends, remember that this electron transport system on one hand shows how the cytochromes are involved at different redox potentials. They are handling over this free electron, and ultimately this electron is released at the end to used for the oxidation of hydrogen proton to convert it into a hydrogen ion. This cytochrome chain, and as the electron freely moving across this is called as an electron transport system, or this is also called as a terminal phosphorylation. Remember, all these cytochromes, they are embedded in the mitochondrial membrane. That is, these particular oxidative compounds or cytochromes, they are present in the inner membrane, inner folding of the cristae or inner folding of the mitochondrion, and all these are embedded in the membrane. So remember, this enzymatic reaction of malate to oxaloacetate, isocitrate to alpha-ketoglutarate, or even the pyruvate to acetyl-CoA, all these reactions are taking place in the stroma or matrix of the mitochondrion, whereas the terminal oxidation, it is taking place in the membrane flow. How it is taking place? There is a diagrammatic representation. I will show you now. You can see that this is the terminal oxidation or oxidative phosphorylation. Now you can see the membrane in between, which is shown by the dark blue color. Below that, you can see the inner membrane space, which is light blue in color. And again, you can see the terminal transport chain, which is present. And this transport chain is nothing but, it's a matrix. Please mute the microphone. Now, you can see here it is clearly indicated the complex 1, the complex 2, the complex 3, and complex 4. Now, the complex 1 is involving FMN, and it is also involving the cytochrome Q. The complex second is involving the only enzyme which is responsible for FADH to FAD. Now, in between, this is coupled by a ubiquinone or coenzyme Q, which is written in the green. Then there comes the complex 3, which is the complex nothing but it is a cytochrome B and cytochrome C. They are again coupled to the next complex by a cytochrome C, and the next last couple is the complex 4, which is nothing but it is a terminal oxidase enzyme. Here you can see that the enzymatic reactions are taking place in the matrix, that is light green in color, whereas the hydrogen protons, they are released in the inner membrane structure, and that's why they are present in the green. At the end, you can see the ATP synthase enzyme. So what is happening, that these hydrogen protons, they are moving across the membrane. When they move across the membrane, they release the energy, and this released energy is trapped by the ADP molecule. It accepts this energy to form an energy-rich phosphate bond, and then it gets attached to the ADP. So inorganic phosphate, which is energy-rich, and ADP, they combine together, and that's why there is a formation of ATP molecule. So all this coupling of hydrogen proton, releasing the energy, trapping this energy by the phosphate, and forming a high energy phosphate bond, and linking this phosphate with the ADP molecule. This is mediated by the enzyme ATP synthase, and from this, with the influx and outflux of the hydrogen proton, there is a continuous generation of ATP molecules. So dear friends, remember, on one side we were discussing about the release of electrons, on one side we were discussing about the release of hydrogen protons, and at the end we have seen that these hydrogen protons are responsible ultimately for the synthesis of ATP molecule. And that's why in terminal oxidation, actually we are discussing two separate parts. One part is a cytochrome chain, which is responsible for the transfer of electrons, and these electrons, they are giving out the energy, and this ultimately energy is used ultimately to trap the hydrogen protons, which is combining with oxygen to form a water molecule. And secondly, the free hydrogen protons, which are released in the inner membrane structure, they are used for the synthesis of photophosphorylation, or they are synthesized for the phosphorylation process. Now remember, as this is involving the oxidation reduction reaction, this whole mechanism of electron transport chain, it is also called as an oxidative phosphorylation. It is also called as an oxidative phosphorylation. It is also called as a terminal oxidation, and it is also called as the electron transport system. So this electron transport system, ultimately it is linked with the phosphorylation process. Now how it is combined with the phosphorylation process, let us come together and discuss this issue also. But before that, we have to summarize what we have learned so far. See, the ATP production in the glycolysis and Krebs cycle, it is written in summary. Now what it shows, In glycolysis, we have seen that the two ATP molecules, they are generated as a net gain. So ultimately 4 ATPs are synthesized and 2 ATPs are utilized. So ultimately we are having the 2 ATPs which are directly as a net gain. Then there is a one oxidation reaction, 3-PGAl to phosphoglycerate, where NADH2 is synthesized. And now we have seen in the electron transport system that this NADH2 is going to generate the 3 molecules of ATP. So as this reaction is taking place twice, we are going to get 6 molecules of ATP. So 6 plus 2, it becomes 8. In pyruvic acid also, 2 molecules of NADH2 are synthesized. And obviously because of that, there is a generation of 6 molecules of ATP. And in TCA cycle, we have seen, or in citric acid cycle, we have seen that at 3 locations of oxidation, the NADH2 is synthesized. So obviously we are going to get the 18 ATP molecules. At one location, there is a FADH2 which is synthesized. So obviously we are going to get the 2 molecules each. So obviously it is taking place 2 cycles. So 4 ATP molecules. And lastly, there is a net gain of 2 GDP molecules also. And these GTP molecules, ultimately they are going to give the 2 molecules of ATP. And that's why in all, when one glucose molecule is completely oxidized to CO2 molecules, we are going to get the 38 ATP molecules. And that's why this is a sort of an energy budget of the complete oxidation of glucose or total oxidation of glucose molecule. The same is written here in a balanced form where ATP are formed, NADH2 is formed, and FADH2 is formed. Ultimately, it is going to generate the 38 ATP molecules. So this is also called as a complete oxidation of glucose molecule, or it is also called as aerobic respiration. As it is in aerobic respiration, a glucose molecule is completely oxidized further. Now, let us study something about the phosphorylation process. Now, you can see how the ATP is working. I have shown the ATP molecule. The ATP molecule is having the 2 components. F0 component, that is the base of the ATP molecule, or ATP synthetase enzyme, which is embedded in the membrane, whereas it is having its head, which is coming out in the matrix, which is free, and it is called as the F1. So F0 and F1, it is a globular structure of ATP synthase enzyme. Now you can see this image on the lower side. You can see the outer side of the membrane, that is the inner mitochondrial membrane, which is written at the bottom, and the F1 particle is seen, that is the Christie particle, which is present towards the matrix. So matrix is nothing but it is the lumen of the mitochondrion, and in between we can see a lipoproteinous membrane, and this lipoproteinous membrane is responsible for transport of hydrogen protons. So you can see the hydrogen protons which are generated in the inner mitochondrial membrane, they are moving across the membrane because of the concentration gradient. Why? Because we have seen that in terminal electron oxidation process or electron transport chain, the hydrogen protons, they are released and they are accumulated in the inner mitochondrial membrane. Now, because of that, the hydrogen proton chains, they are much more higher, and obviously on other side, in matrix, their concentration is low. And so to compensate this, the hydrogen protons, they move across this membrane. Now this membrane is not permitting these hydrogen protons to move across. So there is only one channel or one place that they move across the ATP synthetase enzyme. So they are moving across the ATP synthetase enzyme, and when these hydrogen protons are moving across the membrane, as I shown in the diagram, the ADP is combining with the high energy phosphate bond, and this F1 particle which is hanging in the cristae, it is combining this together or coupling this together to form the ATP molecule. And because of that, it has been found that with every 4 hydrogen protons, there is a synthesis of one ATP molecule. So obviously the balance is that with every 4 hydrogen protons, we are going to generate one ATP molecule at the end. Now, when this diagrammatic representation has to be studied in relation with its mitochondrial placement or location, let us study what we can see. We can see that this is the chemoosmotic hypothesis of ATP synthesis, or it is also called as oxidative phosphorylation. Now you can see that on upper side there is an inner mitochondrial membrane where the hydrogen protons are present. You can see a bilipid layer which is having the lipid molecules as well as the protein molecules. Then in between you can see the channel. Now this channel is nothing but these are cytochromes which are responsible for pumping the hydrogen protons in the inner membrane structure. And then you can see the ATP synthetase enzyme. Now ATP synthetase enzyme, you can see it is having the two components. One component is embedded in the membrane and one component is free, which is hanging. So it is rotating, and that's why it is the embedded portion is called the F0 portion and hanging portion is called as the F1 particle, or that's why it is called as the F1 component which is present on the cristae. Now see what is happening. The hydrogen protons, they are moving across the membrane through this ATP synthetase enzyme. Because of that, the ATP synthetase enzyme, the F1 particle is rotating. When this rotating is taking place, it is combining the energy with the hydrogen protons to the phosphate bond to form the high energy phosphate bond, and this high energy phosphate bond is linking with the ADP molecule to form the ATP molecule. So this movement of hydrogen protons across the membrane and generating the energy and converting the energy into form of ATP molecules, this is called as a chemi-osmotic hypothesis. Now this hypothesis was presented by Peter Mitchell in 1971, and for this purpose he received the Nobel Prize in 1983. So this was the first case where the exact mechanism of ATP synthetase enzyme was shown, the relation of intermembrane structure was shown, and also the correlation of hydrogen protons, pumping of hydrogen protons, and its relation with the generation of high energy phosphate bond was discovered and it was presented. And then because of that, this was the first evidence where it was proved that how the electron transport scale, which is releasing the electrons, which is releasing the hydrogen protons, they are responsible ultimately for the generation of ATPs. And that's why this is also called as a terminal or oxidative phosphorylation. So we started our discussion with the glucose molecule. We have seen that the glucose molecule at intervals it is getting oxidized. When it is getting oxidized, the cofactors like NAD and FADH2, they are getting reduced to NADH2 and FADH2, and they are ultimately passing through the electron transport chain where they are releasing the electrons and also hydrogen protons. And lastly, we have seen that how these hydrogen protons and the electrons, they are responsible for generation of not only ATP molecule, but the hydrogen protons and electrons, they are responsible for the formation of water molecule. For this purpose, I want to take you just one slide back, and you can see the complex 4. Now this complex 4 is nothing but it is a terminal oxidase enzyme. Now this terminal oxidase enzyme is known as a cytochrome A and cytochrome A3 complex, which is containing the iron as well as it is containing the copper. And the main function of this enzyme is combining the free electrons with the hydrogen proton to form the hydrogen ion. And here this hydrogen ion is joining with the atmospheric oxygen molecule. And here this oxygen molecule, ultimately it is getting hydrolyzed. It is getting hydrolyzed into a water molecule. And that's why, dear friends, this electron transport system or terminal oxidation is very important sequence of reactions which is ultimately generating and giving rise to the H2O molecules, but it is undergoing oxidation reduction which is further used for energy transfer, and this energy is ultimately used for the synthesis of ATP molecules. And because of that, remember, as such a type of ATP synthase enzyme is present in the mitochondrion, with every glucose molecules, it is synthesizing and producing the 38 ATP molecules, and large number of glucose molecules, they are being oxidized in respiration process. Because of that, the mitochondrion are known as the powerhouse, because they are continuously supplying the ATP molecules to the whole cell, not only to the mitochondrion, but they are supplying the ATP molecules even to the cytoplasmic reactions. And because of that, the cell is undergoing the different metabolic activities. So because of that, all this process of terminal oxidation and synthesis of ATP molecules through oxidative phosphorylation is very important. So the essential components required for the chemiosmosis are the proton pump, the proton gradient across the membrane, and ATP synthase enzyme. So proton pump is nothing but it's the F0 component of the ATP synthase enzyme, which is pumping the hydrogen protons across the membrane. The ATP synthase is an enzyme which helps in ATP synthesis, and the enzyme, as I told you, has got two parts, F0 and F1. The F0 is the part which is transporting the protons across the membrane as a transmembrane channel or canal, while the configuration changes in the F1, which is activating the enzyme. They are phosphorylizing the ADP, that is, they are phosphorylizing means they are adding one more phosphate bond here to add one more phosphorus group to the ADP and converting this ADP into ATP. And that's why there is a synthesis or generation of ATP molecule in the matrix of the mitochondrion. Remember this. Now the proton gradient, which is developed across the membrane, is the driving force or is the activating force for the ATP synthase. Now at the end, what is the significance of the respiration? It is the energy releasing and supplying process in all living organisms. It also converts the food energy into metabolically usable form of the chemical energy. So the reserve for energy, which is present in the form of reserve food material, it is ultimately converted into the ATP form. It is releasing the energy in a controlled and stepwise manner, as a result of which most of the released energy is properly utilized for various cellular activities. Now these activities like metabolism or cell division, obviously the growth, and some of the energy obviously during this formation is being lost as a heat. The respiration converts the energy very efficiently. Out of the total 673 kilocalories of the energy released per molecule of the glucose, 456 kilocalories of the energy is converted as through the 38 ATP molecules. So this is about 66 to 68% of efficiency of oxidation. Various intermediate products of glycolysis and Krebs cycle, most of these products, they are organic acids. These are used for the biosynthesis of other complex organic compounds in the cell metabolism. Ultimately, the carbon dioxide required for the photosynthesis by the green plants, it is replenished by the carbon dioxide released during the respiration. Ultimately, when the glucose molecule is broken down, it is broken down in the form of oxidase form, and that's why it is released as a CO2. And this CO2 ultimately is accepted by the photosynthesis membrane or by the chloroplast to regenerate the glucose molecule. Thus, the photosynthesis and respiration are complementary to each other. But remember, respiration is a universal physiological process which is present right from the bacteria to all living world, including plants and animals, whereas the photosynthesis is present only in the plants and also in green organisms. It also helps to maintain the balance of the oxygen and carbon dioxide in the nature. Fermentation, that is anaerobic respiration, which is a type of anaerobic respiration, is helpful in the industrial production of various useful products such as alcohol, antibiotics, vitamins, organic acids, bakery and dairy products, and also during the tanning process where the skin is converted into tannin leather. And this is the way the respiration, which is important from the physiological point of view. We have discussed it in detail through its proper mechanism, and we have tried to understand this whole mechanism in a stepwise manner. Now, I'll be providing you all the detailed notes of this respiration on your WhatsApp group. So study well, prepare your own notes. Remember, there are three components which are very important from examination point of view. The first component is glycolysis. The second component is TCA cycle or citric acid cycle or even Krebs cycle. And last component is obviously the ETS or electron transport system. But nowadays, many of the times the examiners are much more interested in asking the details of terminal oxidation or oxidative phosphorylation. Remember, oxidative phosphorylation is present in the mitochondria, whereas the photophosphorylation is present in the chloroplast. Oxidative phosphorylation is generating the ATP molecules by the oxidation of NADH2, which is present in the mitochondria, whereas the photooxidation is taking place in the chloroplast, and therein the NADPH2 is involved. So NADH2 is present in the mitochondria and NADPH2 is present in the chloroplast. So remember, even though in both the cases the ATP generated in oxidative phosphorylation, the NADH2 is involved. In phosphorylation or photophosphorylation, the NADPH2 is involved. So examiners are much more also interested in asking a question on terminal phosphorylation or oxidative phosphorylation. So I do request you to kindly prepare yourself, prepare your own notes, check it out, show the notes to the either Patil ma'am or to me. I will go through the details and make a comment. Prepare yourself well for this topic because this is one of the most important topic as far as the physiology is concerned. And then read the books. The books are available, the notes are available. Go through the notes, prepare yourself well in advance. And with this, we will stop today with the end of respiration discussion. Now we'll be going ahead with the last component of our discussion, and that is translocation. I'll be taking this translocation in 2 lectures on coming Friday and Saturday. So we'll be having a gap of 2 days, and on 26th and 27th, I'll be completing the remaining component of physiology, that is the translocation. I wish you all the best of luck. Prepare good notes, get it checked, read out, and prepare yourself. Wish you all the best. If at all you are having any comments, if at all you are having any doubts, if at all you are having any queries, do ask me through the chat box. Otherwise, if there are no any queries, record your attendance